Hi, and welcome to today's Oxford Centre for Personalised Medicine Flash Interview vlog, where we talk to different people involved in the field of personalised medicine about their life, their work and their experiences. Today, we're very fortunate to be joined by Professor Simon Leedham, Professor of Molecular and Population Genetics in the University of Oxford, and also an honorary consultant gastroenterologist. Simon, it's a privilege to have the opportunity to talk to you today. Would you be able to kick off by giving us a brief tour of your background?
Hi, Catherine. Thanks very much for the invitation to do this really nice series. As you mentioned, I'm a clinician, so I trained in medicine and I specialised in gastroenterology. And I did a number of years of junior doctor training and then got to sort of registrar level, which is the mid-grade doctor level, and realised that I really wanted to spend some more time exploring something else, something in a bit more depth, something that interested me. So I was lucky enough to get an MRC funded PhD and went to work with a chap called Professor Sir Nick Wright at the London Research Institute in London and did a fabulous project looking at clonal evolution in intestinal lesions. And I think that really kind of changed my life, really. It changed my career trajectory because it meant that I didn't want to be a medical doctor anymore. I think I wanted to focus on the science. And then I did that, and sort of over a series of grants, a clinician scientist post brought me to Oxford about 12 years ago. And it was my privilege to direct the Centre for Personalised Medicine for 5 of those. My research interests, I guess, are in the gastrointestinal stem cells. So the stem cell is a cell that lives at the bottom of all of the intestinal crypts. And we're really interested in how that cell behaves and the signaling pathways that control them. Because it's stem cells that are thought to be the cell of origin, both of cancers and also those sorts of particular cells that we're really aiming to kill with our cancer therapies.
Lovely. So what does the term personalised medicine mean to you then?
Yeah, great question, and one that I sort of struggled with during the time as director of the CPM, because it's such a broad term, and I think it can be interpreted by many different people in many different ways. But personally, I like to think of it couched in the term of it's trying to give the right patient the right medicine at the right time. But how we do that really is both a challenge and also a number of opportunities. There are a number of arms that you can use, such as population health or broad epidemiological studies to stratify patients into particular groups. So having a way of understanding how different patients are subgrouped. Another way is to look at individual patients and then use molecular and genomic tools to help to characterize their disease, so to understand their disease in a lot more detail. And then the way that I'm particularly interested in personalized medicine is looking to use drugs in a biologically informed way. So trying to address a signaling pathway that we know to be disrupted, a signaling pathway we know goes wrong, and then trying to find a drug or use a drug that will manipulate that pathway and help the patient that way.
So focusing in then on personalised medicine and cancer a bit more, where are we at the moment, and how specifically does your research on gastrointestinal cancer fit into that field?
Yeah, great question. So I think for many years, cancer therapies have focused almost exclusively on trying to kill the cancer cell itself. So cancers are hugely complex organisms as such. We've had some success in directing cancer treatments to the cancer cells by measuring the genetic mutation burden in those cells and then directing our drugs at those mutations. Now, these therapies are called targeted therapies. And although we've had some notable successes with that, there's also been some notable failures, and perhaps the range of success hasn't been as broad as we might have hoped. One of the problems of this is that tumours tend to develop resistance to those sorts of drugs. And I think on the back of this, we're really starting to shift our focus a little bit, not just looking at the cancer cells and the cells, but also at the other cells in the cancer. So as I say, the cancer is not, cancer cells don't exist in isolation. They're complex ecosystems where the cancer cells are supported and surrounded by huge numbers of other cells. Now, that may be blood vessel cells or stromal cells or even a constellation, a whole range of immune cells. So we're particularly interested in how these surrounding cells, which we sort of group together as calling the tumour microenvironment, how those surrounding cells support and regulate and can sometimes even control the cancer cells themselves. And we think that if we attack not just the cancer cells, but also the supporting microenvironmental cells, then we might be more effective at killing cancer cells off. And I guess the best example of that is immunotherapy. So immunotherapy is a new style of treatment that's really come on stream in the last 5 to 10 years. It's really been the greatest therapeutic innovation in cancer in 20 years, I would say, in a generation. And what this does is it harnesses the power of the immune cells that are circulating within a tumor and gets them to attack the cancer cells themselves. So it's really harnessing the power of the microenvironment to control, regulate, and even kill the cancer cells. And I think that's really where my interests lie at the minute.
So what are the biggest challenges then that we're facing in terms of personalised cancer medicine, and how are we going to overcome them? And have you got any kind of success stories that you can share?
Yeah, great question, which I think alludes to what I've just commented on in terms of resistance to drugs. So cancers, like everything, are really subject to the laws of Darwinian evolution. Now, this means that cancers, like an animal species, for example, the finches in the Galapagos Islands, are capable of adaptation to new environments. And it's this capacity for adaptation that drives resistance to the drugs that we use. And this leads to relapse in our patients. So what do I mean by that? Well, what I mean is that tumours are really rather diverse. They're molecularly and phenotypically diverse. And that's called tumour heterogeneity. And it's this heterogeneity, this diversity, means that an individual therapy is very unlikely to be effective in killing all of the cancer cells at once. So that means if you leave some cells behind, the cells that are resistant to those therapies are selected for, just like natural selection in evolution. And it is those cells that then repopulate the tumor. Now, often that adaptation isn't particularly visible to our diagnostic tools. We can't measure that adaptation with a CT scanner, for example, but we can detect these adaptive shifts in the tumor in response to therapies using molecular tools. And we've recently spent a lot of time developing a particular molecular tool that can identify a responsive shift in the appearance of the tumor in response to a selective pressure. And by selective pressure, I would mean like a drug treatment. We think we can use these molecular tools to measure the impact of treatments in cancer patients. And if we can measure the effect of a therapy, we think it will help us to decide more cleverly, more biologically, which therapies to use and which order to use them in order to make sure we kill as many cancer cells as possible.
Great. So where do you see the future of this going then, maybe in the next kind of 5 to 10 years? And what do we need to achieve those goals?
Yeah, so I think the achievements of the last 20 years are extraordinary. The awesome power of the technological tools that we've now got at disposal is extraordinary. I mean, we've got tools that can measure DNA and RNA in a single cell. We've got new platforms that allow us to assess cell interactions on a slide in a single section. And this has really driven a revolution in our understanding of cancer biology. And I think it's this understanding of biology that's really gone exponentially upwards since the human genome was sequenced in the 2000s. This increased understanding is now the driving force behind the development of drugs, biomarkers, and all the advances that we now have. So it's biology that's driving advances. So I'm hopeful that particularly the understanding of the rules that govern that tumor ecosystem that I just talked about, the idea that a tumor is not a cancer cell in isolation, it exists in an ecosystem. I think if we can understand the rules that govern the cellular interaction between the cells in that ecosystem, will allow us to develop a whole new generation of drugs that will manipulate those cell interactions. And immunotherapy was the example I gave before, and it's a great example of that. That's using a drug that harnesses the power of the immune system to kill cancer cells, not touching the cancer cells themselves as such. So I'm also really encouraged by the drive towards cancer early detection and prevention. That's another exciting advance in the last 5 years or so. The improvement in cancer detection means that most patients' tumors are diagnosed earlier, which means they're easier to treat, and there's a much higher chance of survival. And I think there are huge opportunities around the idea of precision or personalized prevention in this space as well. And what I mean by that is if we can identify the disrupted pathways in patients at the very earliest stage of cancer initiation. So even in a pre-cancer setting, perhaps we can develop drugs or maybe even cancer vaccines that will help attack those cancer precursors in situ in a patient before they develop cancer in the first place. So I think there's a huge amount of excitement and opportunity, and that's notwithstanding the challenges that cancer provides us with its complexity. But I think we do have the opportunity and the exciting opportunity to make advances in the next few years based upon the technological advances that we've made in the last 20.
Fabulous. It's been an honour to talk to you today. Any final comments you want to make? Any words of wisdom for people in wanting to go further in the personalised medicine field?
Well, I would just sort of comment on your fabulous interview series that you've developed here, Catherine. I think it's a really lovely series, really fascinating interviews. I'm hugely encouraged about the ongoing successes of the Oxford Centre for Personalised Medicine. I think words of wisdom would be that if there's anybody out there that's interested in personalised medicine, then look out for when the Oxford CPM fellowships come up, because you're getting fantastic people doing fantastic things. And thank you very much for the opportunity to talk to you.
Brilliant. Thank you very much.